is full of life, but planets like Mars, not so much. But, as scientists have recently shown, one mighty moss may manage Mars. Let's learn about Centrichia cananervis. S. cananervis is a desert moss found around the world, from California's Mojave Desert to Antarctica to deserts in northwest China where temperatures range from -40 to 65 degrees Celsius, or -40 to 149 freaking degrees Fahrenheit. Simply by virtue of the environments it thrives in, S. cananervis is a tough little moss. So, if ever an Earth plant could make this relationship work with Mars, S. cananervis should be the moss to do it. And in this article in the Innovation, a group of botanists put S. cananervis through its paces to test whether tough, by Earth standards, could be enough to withstand Mars' unforgiving environment. First, a review of the ways that the Martian environment is different from Earth's in ways that, shall we say, discourage Earth life from taking hold. Mars on its best day may pull down temperatures of 20 to 35 degrees Celsius, or 80 to 95 Fahrenheit, but most of the time it's way VF colder. The mean surface temperature is estimated somewhere around -63 to -55 degrees Celsius, -61 to -81 Fahrenheit, so even our mega-resilient moss would be tested under these conditions. Second, the atmospheric surface pressure of Mars is less than 1% of Earth's, so thin that it precludes liquid water on its surface. Then there's the composition of that atmosphere, where Earth's atmosphere is about 78% nitrogen, with about 21% oxygen, and the remainder mostly argon, and like 0.03% CO2. What little atmosphere Mars has is overwhelmingly CO2 at 95%. With roughly 2% nitrogen, 2% argon, and only 0.15% oxygen. Yes, plants love themselves some CO2, but they also rely on nitrogen from the atmosphere, which either they fix into bioavailable format themselves or with help from symbiotic bacteria. So with a tiny fraction of Earth's atmosphere and disproportionately less nitrogen, yeah, not ideal. And if our moss survives the temperature conditions, the minimal humidity, the thin atmosphere, and the different composition, there's still, the radiation. With so little atmosphere and no global magnetic field, Mars' surface has minimal protection from cosmic and solar radiation, and there's no SPF 45 going to make up for that. Some aspects of Mars' environment have no counterpart on Earth. Nothing here is going to prepare you for 1% of your usual atmosphere, and most of that being carbon dioxide. But S. cananervis is somewhat adapted to some of these conditions. The temperatures on Mars are even more extreme than on our moss's home turf, but at least S. cananervis has some overt thermotolerance adaptations. Ditto humidity. It's a desert moss, and it acts like it. S. cananervis has specialized twisted leaves that minimize its surface area to reduce water loss, and its cells remain intact even when completely dehydrated. Together, these features allow S. cananervis to survive big swings in temperature through freeze-thaw cycles, as it might have to in Earth's spring in some of these areas, and that could help it survive Mars' swingy temperatures as well. Other conditions aren't directly relevant on Earth, but our moss's adaptations may still help. S. cananervis is quite UV tolerant, and there's a high UV index out there in the desert, and has a particularly robust oxidative stress response that kicks in to help it survive UV radiation exposure. UV isn't the sole or even primary electromagnetic frequency we're worrying about with cosmic and solar radiation, and most UV isn't ionizing radiation. But if you're a plant on Mars, the radiation you're going to get hit with is going to induce an oxidative stress response, so better to have the adaptation than not. Will it be enough? Sometimes you just gotta test and find out, and this paper is about putting S. cananervis through a battery of tests to figure out if it's even worth trying to send Earth plants to Mars, because if anyone can, it's this kiddo. So, How did it do? First, dehydration. This is a bit of an underhanded softball pitch to a desert moss, and to no one's surprise, Escananervis absolutely crushes it. After losing 99% of its water content over 40 minutes, our researchers rehydrated the shriveled, blackened moss. Within 2 seconds of rehydration, Escananervis turned green again. Within 20 seconds, its leaves recovered their shape, and by 2 minutes, the planet's water content was fully restored. When desiccated, the moss's photosynthesis was essentially shut down, but its photosynthetic activity came back online within 2 minutes as well. So, would Escananervis thrive in a perpetually desiccated state? Perhaps a strong word, but it can easily bunker down until conditions improve, and it can quickly take advantage of any moisture that does come its way. Now, temperature. The highs on Earth in Escananervis's environment are warmer than anything it's likely to experience on Mars, but the lows are far lower. Using both dehydrated and fully hydrated moss, our researchers tested temperature tolerance in 2 ways. Long-term freezing for 3 to 5 years in a minus 80 Celsius freezer, and 15 to 30 days in negative 196 Celsius in a freaking liquid nitrogen storage tank. Let no one say they're going easy on this poor plant. After the freezing period, they thawed the plants to see if the moss could regenerate and grow. And hey, did the frozen mosses outperform the control moss that just got to live in a nice room temperature lab? No. But both mosses stored for weeks in liquid nitrogen and years in a minus 80 freezer were able to recover and grow, regenerating new branches first slowly, then at a comparable rate to controls within 1 to 2 months. Interestingly, the pre-dried moss outperformed the ones that went into deep freeze fully hydrated, which could be a function of the water itself, or that the drying induced gene expression that made the moss more resilient in ways that also applied to freezing. From here in our tests, though, we are leaving Earth-like conditions, and our botanists just clobber this Escananervis with gamma radiation to test its resistance to ionizing radiation. Because scale is important here, I'll tell you that about 0.75 gray of ionizing radiation is enough to cause acute radiation syndrome or radiation sickness within minutes to hours. And 0.75 gray is about 18,000 chest x-rays worth of radiation all at once. Oh, but we need to test Martian levels of radiation, our scientists say. The bidding will begin at 500 gray. 500 gray. And then a dose response curve up to 16,000 gray. Again, we test both desiccated and hydrated Escananervis moss, expose them to the insult, in this case radiation, and then test their ability to recover, regenerate, and grow. And hey, did the radiation-exposed mosses outperform the control moss that just got to live in a nice, non-irradiated lab? No. Oh, actually, kinda. Moss exposed to 500 gray of gamma radiation had a significantly higher regeneration rate than control moss for the first couple weeks of recovery, and by 2 months post-exposure, both 500 and 1,000 gray exposed moss actually had significantly more newly growing branches than controls. In fact, moss exposed to up to 4,000 gray of gamma radiation ultimately regenerated at least a little within 2 weeks of recovery. Although, yeah, above that, you've still just got some dead freaking moss. Once again, though, the desiccated mosses were the better performers, with the hydrated mosses dying at lower doses of radiation with less exposure time than their dried-up counterparts. So, we're good for drying, freezing, radiation. Now we gotta put it all together and test our plants in the Mars simulator, i.e. the Planetary Atmosphere Simulation Facility, or PASF, at the National Space Science Center at the Chinese Academy of Science. Basically, they toss moss in the big tabletop chamber. The gas tank on the right's kind of hint to the scale we're looking at here. And this can simulate the low pressure, swingy temperatures, and atmospheric composition of Mars. The radiation in the PASF is approximated with UV at non-ionizing frequencies, so that part's not the closest approximation. But since we just saw S. cananervis suck down 1000 gray and ask for seconds, if it can't handle the radiation exposure on Mars, we certainly couldn't. So, how did our moss do after a week on Mars? Once again, the desiccated moss recovered more effectively. The fully hydrated moss didn't even like a day in the Mars tank, although they did regenerate after exposure to Mars. They were pretty sulky about it. Even dry moss that spent more than a couple days in the Mars tank took a couple weeks to recover from the experience, but they did ultimately grow new branches and return to a healthy regeneration rate comparable to control plants. As the drying study showed, desiccated S. cananervis has minimal photosynthetic activity. So while the Martian atmospheric composition didn't actively seem to kill the moss, the dried out moss wasn't really all that metabolically active at the time, so it's not like it was drawing resources from the environment too much one way or the other, most likely. Overall, I wouldn't say that S. cananervis wants to put Mars up as its next vacation destination, but this study indicates that the desert moss could certainly live there, making it the first plant that could plausibly survive, and joining the ranks of some bacteria, algae, and lichen that can probably also survive the red planet. That said, it's hard to say whether the moss would thrive. After all, this moss could spring back to health after exposure to these Martian conditions, but there's no after on Mars. There's just more Mars. In fact, this whole go dormant and survive thing that S. cananervis has going on reminded me a little of tardigrades with like the desiccation, the temperature, the radiation tolerance. Like those microanimals, S. cananervis isn't an extremophile that's actively adapted to specific extreme conditions, but they are both extremotolerant, capable of withstanding a range of extreme conditions and kind of muddling along until things get better. And if these two species share that feature, it's because tardigrades play a role on Earth that this study's researchers hope S. cananervis could perform on Mars, the pioneer species. On Earth, environmental disasters can periodically wipe out an ecosystem. Think wildfires, floods, lava flows, and so on. Those ecosystems have to build back up, usually starting with bacteria and with lichen that can break down rocks for plants to take hold in. Moss, like our S. cananervis, are usually among the first plants to take hold in those rebuilding ecosystems, and tardigrades in those ecosystems eat bacteria, lichen, algae, and yes, moss. Pioneer species are resilient by definition. They're the ones that can survive conditions other species can't. But this ecological role also affords an advantage to those species. Because they're so resilient, they can feast on whatever resources are there without less resilient, but perhaps more specialized, competitors. In this primary succession after ecological disturbance, as the population of pioneer species grows and turns over, the dead biomatter from those first generations contributes to nutrient-rich soil, which eventually supports a second wave of species that can take hold as the environment starts to normalize. That's when you get vascular plants like grasses and shrubs, or little animals that eat the pioneer species, and so on. Many pioneer species may be supplanted at this stage, though others continue to provide valuable ecosystem services to vascular plants and all the way up the food chain. And on Earth, mosses are particularly important for this, both their support of the plant species and their contribution to what's called the biological soil crust, the ground cover found in arid lands that improves its water retention and provides structural stability, carbon, nitrogen for all those other life things that want to live. So, if S. cananervis really can withstand Mars, and they're joining bacteria and algae and lichens, You can think of the Martian environment as one big ol' ecological disturbance, and these as a squad of pioneer species whose signature trait is their resilience. And yes, I'm sure some folks are thinking about the implications of those ideas in a terraforming context. The discussion section of this paper certainly was. But this doesn't have to be about giving the whole planet of Mars a full biological soil crust makeover. When packing your suitcase for interplanetary travel, where every kilogram is precious, there's a lot to be said for identifying species that can contribute to generating nutrient-rich soil once we're on Mars. Now, whether or not it can feasibly serve that purpose, that remains to be seen. After all, S. cananervis can survive simulated Martian conditions by going into dormancy, but this moss can't contribute to its local ecosystem unless it wakes up occasionally, does some photosynthesis, and otherwise interacts with its environment. In the meantime, though, there may be things we can learn from S. cananervis that could be applied to other plant species to enhance their resilience. I'm not saying we can splice in some moss genes and make carrots as drought-resistant as a desert moss. After all, those resistances come from multiple attributes, including their leaf shape and so on, and those things aren't going to apply to spinach. But if humans are ever going to survive on Mars, plants are too. And this moss may be our best first chance.